Assalamualaikum warahmatullahi wabarakatuh. Hello everyone. Before we begin, let me introduce myself briefly. I'm Aulia and I'm a student at Universitas Negeri Semarang. In this occasion, I will present the result of a laboratory work report about the evidence of peroxidase enzyme. First of all, I'll explain what is the peroxidase enzyme and how to prove the existence of peroxidase enzyme. I'll continue by showing three samples containing peroxidase enzyme and why there is a difference in result when reacted with H2O2 hydrogen peroxide solution. To finish, I'll be concluding the result of the experiment. So, do you know? The liver is a very important organ in our body. One of the roles of the liver is to neutralize toxins that enter the body either through food or drink by enzymes found in it, one of which is the peroxidase enzyme. Imagine if we didn't have a liver, who would protect our body if we ate something toxic? So let's discuss more about the existence of peroxidase enzyme. Introduction. Peroxidases are enzymes that catalyze oxidation reaction by hydrogen peroxide from a number of substrates, which are hydrogen donors such as phenolic compounds, aniline, pyrrolidinol, and ascorbic acid. Peroxidase enzymes play a role in decomposing excess hydrogen peroxide into water and oxygen. It's found the liver and peroxisome in plants. Besides coming from the body directly, this enzyme is also found in food sources such as broccoli stalks, corn stalks, and white radish. And the purpose. This experiment aims to prove the existence of peroxidase enzyme in the sample. Let's move on and let's discuss about the experiment procedures. The method used is the experimental method. There were three samples tested: 1. Liver of chicken solution, 2. Papaya leaf solution, and 3. Turmeric solution. The material used is H2O2 or hydrogen peroxide solution. The experiment procedures. First, prepare 3 clean and dry test tubes. Second, grind all the excess ingredients first. Third, treat them as shown the next table. Tube 1. Fill 1 milliliter liver of chicken solution and 5 drops H2O2 solution. In tube 2, fill 1 milliliter papaya leaf solution and 5 drops H2O2 solution. And tube 3, fill 1 milliliter turmeric solution and 5 drops H2O2 solution. And then close the end of the test tube immediately after adding the H2O2 solution or hydrogen peroxide solution. Then observe the bubbles formed. The last step is put the coals into the test tube and then observe the embers is on or off. Now, this is the results of the experiment. We can compare in each tube, tube 1, tube 2, and tube 3. In tube 1, there are a lot of bubbles formed, plus, and in tube 2, it's not as much as tube 1, plus, and in tube 3, it's not as much as tube 1 as tube 2, only plus, but the embers is on in 3 tubes. So why there is a difference number of the bubbles in this experiment? Let me go into some more detail about why is there a difference in the number of bubbles in 3 samples. Tube 1, the liver of chicken solution reacted with H2O2 solution to produce lots of bubbles, plus, and the coals ignited. This shows that liver of chicken contains peroxidase enzyme, which are indicated by the presence of air bubbles. These air bubbles indicate the presence of water resulting from the reaction of the two solutions. These air bubbles also contain oxygen, so when exposed to hot coals, they will ignite. Based on the experimental result, the liver of this chicken contains the highest peroxidase because the function of the liver is to neutralize toxins. Therefore, the most peroxidase produced compared to other samples. In tube 2, the papillip solution reacted with the hydrogen peroxide solution to produce quite a lot of bubbles, and the coals ignited. The reason of the coals ignited is same with previous tube, and the difference in the number of bubbles formed in tube 2 because in plant, this enzyme is produced by peroxisome. Peroxisome work is influenced by photorespiration. The higher the photorespiration, the higher the peroxisome activity. In tube 3, the turmeric solution reacted with the H2O2 solution to produce a few bubbles only plus and the coals ignited. The reason of the coals ignited is same with previous tube in tube 1 and in tube 2, but the difference number of bubbles because the turmeric located underground, so the photorespiration rate is low. This is the reason why the amount of peroxidase enzyme produced by turmeric is not as much as that produced by the liver of chicken and papaya leaves. This is the documentation of the experiment. We can compare the bubbles formed in tube 1, tube 2, and tube 3. In tube 1, there are a lot of bubbles formed, and in tube 2, the bubbles formed is not as much as tube 1, and in tube 3, the bubbles formed is not as much as tube 1 and tube 3. Let's review the key points in this session. The liver of chicken, papaya leaves, and turmeric contain peroxidase enzyme. However, the liver of chicken has the highest peroxidase enzyme and evidenced by the large number of air bubbles produced, then followed by papaya leaves, finally turmeric. The liver of chicken contains many peroxidase enzyme because one of the functions of the liver is to neutralize toxins. And the last one is acknowledgement. I just want to say thank you to those who have helped me in preparing the presentation draft regarding peroxidase enzyme. Special thanks to laboratory assistant, lecturers, laboratory technicians, and all my friends. That's all from me. I'm sorry if there are any mistakes and thanks for your attention. Wassalamualaikum warahmatullahi wabarakatuh.